So now we are on to another group of organisms, and we're still talking about gram-negative bacilli or gram-negative rods. And so you can see that there are a lot of different organisms that are gram-negative rods. So there are a lot of organisms to memorize, especially for this second exam. Now this group of organisms that we are going to discuss is different in some of the other groups because the previous group of gram-negative rods we talked about were in the Enterobacteriaceae family. Well, the organisms we are going to discuss today are also gram-negative rods, and some of them also can cause intestinal infections, like some of the Enterobacteriaceae members. However, none of the organisms in this group are Enterobacteriaceae family members, so it's a very important distinction that you have to know. This lecture has been broken up into 3 different parts, so we are going to start out right now with part 1. Now the other fermentative gram-negative rods, in addition to the Enterobacteriaceae members are fermentative. They all ferment glucose. Well, the organisms we are going to discuss today are also fermentative. They will ferment certain carbohydrates. And the genera in the fermentative gram-negative rods are Vibrio, Aeromonas, and Plesiomonas. And we're going to start out by talking about the genus Vibrio. Now there are numerous Vibrio species, and the ones that we are going to discuss, of course, are those that are medically relevant. And the species that we are going to talk about in more detail are Vibrio cholerae, Vibrio parahemolyticus, Vibrio vulnificus, and Vibrio alginolyticus. In general, the Vibrionaceae family has 30 species, and there are 12 overall that can infect humans. All of these organisms are commonly found in water or aquatic environments, and they cause a variety of diseases, from gastrointestinal illness to wound infections. Microscopically, these organisms are non-spore formers. They can be numerous, morphic shapes, so they can have slightly varied shapes to them. They are described in textbooks as curved gram-negative rods. You may only see that curve from a gram stain of a clinical specimen, though. If you take a Vibrio organism, streak it out on an agar plate, pick up a colony and gram stain it, it's just going to look like any other gram-negative rod. You really wouldn't be able to differentiate a Vibrio from another gram-negative rod, which is why the biochemical tests among the gram-negative rods are so important. The Vibrionaceae members possess a polar sheathed flagella when they are grown in a broth, and they produce peritrichous unsheathed flagella when they're grown on solid media. Nevertheless, they are motile. They do have flagella regardless of what type of media you grow them in, so they definitely are motile organisms. Here's a gram stain of Vibrio, and if you really look carefully, you could see a slight curve to some of the rods, but it is not very distinct, and it wouldn't really be enough to be able to diagnose an infection if you looked under a microscope. These organisms are facultative anaerobes, so they grow pretty much anywhere. And here is one of the reasons why they are not members of the Enterobacteriaceae family. You guys all remember that in order to be a member of the Enterobacteriaceae family, you have to be oxidase negative. These organisms are oxidase positive. You must know that. You must know the difference. There will be case studies on the next exam. There will be case studies on the final exam. And sometimes that is the only thing that will allow you to differentiate one organism out of a list of organisms, is just this oxidase reaction. These organisms are able to reduce nitrate to nitrite. Most of the Vibrio species are susceptible to the Vibriostat disc, or the O129 Vibriostatic compound. And this is a little disc, just like the antibiotic discs, that contains 2,4-diamino-6,7-diisopropyl-pteridine. And you drop that on your lawn of organism, incubate it, and you are going to look for a zone of inhibition or no zone. Now, most Vibrio species are susceptible, so there will be a zone of inhibition around the disc, but not all of the species. So that's when this disc becomes important. It's one of the features that will allow you to differentiate one Vibrio species from another. Most of the Vibrio species exhibit what's called a positive string test. So if you drop 0.5% sodium deoxycholate on a colony growing on an agar plate, the colony will become very mucousy and stringy. So if you took a sterile loop and you started to pick at that colony, it would be very stringy. Most Vibrio species are halophilic. Halo meaning salt and philic meaning loving. So most Vibrio species can grow in a high salt concentration except for Vibrio cholerae. Now that is very important. So that's one of the distinguishing features that you need to know. So here we have Vibrio cholera and non-cholera Vibrios. So what you do is, let's say you thought you had a Vibrio, you make a suspension, you make a lawn of growth on a just a regular nutrient agar plate. You drop an O129 susceptibility disc, and on the left you see that that organism is resistant. It grows right up to the disc. On the right, that organism is susceptible. There is a zone of inhibition around the disc. So all Vibrio species are susceptible to this vibriostatic disc. So we're going to talk a little bit about each one of the Vibrio species. So we have Vibrio cholerae, O1 and non-O1. This group of organisms cause intestinal infection. They cause what's called rice water stool. And so what cholera, the disease cholera, is characteristic for is a very, very watery stool that has little flecks of fecal material and mucosal epithelial cells in it. And it kind of looks like little bits of rice in water. It is transmitted through ingestion of contaminated food and water. Vibrio parahemolyticus causes severe cramping and a watery diarrhea, and the common transmission is ingestion of raw shellfish, especially oysters. Vibrio vulnificus causes both gastrointestinal infections as well as wound infections. It has been associated with causing bacteremia, and this is due to ingestion of contaminated food as well as just being in contact with contaminated water. So someone that has an open wound in their, let's say their foot or their leg, and they're wading through contaminated water, the organism can cause a wound infection that way. And then Vibrio alginolyticus is the least pathogenic Vibrio species that we are going to talk about. We're going to start out, as we always do, with our bad organism, our most virulent organism, and out of the Vibrio organisms, it's Vibrio cholerae. Vibrio cholerae causes the disease cholera. Cholera is also known as Asiatic cholera or epidemic cholera. Cholera is an endemic disease in developing countries, and Vibrio cholera 01 is the pandemic strain. Cholera is a very rare reported disease in the United States, but in other areas of the world, it is a very serious problem. So in this slide, you could see the worldwide cholera outbreaks, and this is in 2009. So you can see that in the United States, up in Canada, you don't really see much cholera, but in other areas of the world, Southeast East Asia, some areas of Africa, few areas in South America, they do occasionally get these terrible outbreaks of cholera that kill many people. So what cholera does is it causes an acute diarrheal disease. Again, it's transmitted through ingestion of contaminated water is the most common cause of infection. Usually this is due to improperly preserved or handled food. So in other words, if you go to a developing country, you only want to drink bottled water that you know comes from a clean source, and you don't want to eat any foods that you aren't sure how they were handled. You also don't really want to eat raw types of foods like salads that might have been washed with contaminated water. So you really have to think carefully when you're traveling to another country of exactly what you're going to eat and what you're not going to eat, because sometimes something that doesn't seem like it would be a problem can be. It causes severe gastroenteritis with vomiting and diarrhea. Stools are described as rice water. Again, you get your, flecks of mucus and epithelial cells, little bits of fecal material with this very, very watery diarrhea. What happens is people lose so much water, they're rapidly losing fluids, so they get an electrolyte loss, imbalance in their electrolytes, which leads to dehydration, which can then lead to hypovolemic shock, metabolic acidosis, and can cause death within hours. That is how severe this diarrhea is from cholera. The virulence factors produced by Vibrio cholerae, the organism can produce an enterotoxin called cholera toxin or choleragen. The organism colonizes the small intestine. Once it colonizes the small intestine, it produces a toxin. So Vibrio is an infection. It is not a food poisoning, as we've discussed previously, because you actually ingest the organism gets into the small intestine, and then it starts to produce the toxin. The toxin has two subunits. Subunit A and subunit, there are 5 binding B subunits. And what happens is you get an accumulation of cyclic AMP, which stimulates the hypersecretion of electrolytes and water out of the cells, which is why you have this massive watery diarrhea. So here's a depiction of cholera toxin. I don't expect you to know the mechanism of cholera toxin. If this were a pathogenesis course or infectious disease mechanism course, I would expect you to know the mechanism of action, but I just included this here for anyone who might be curious on how this toxin works. Now, treatment or management of a person with cholera, you have to administer intravenous or oral fluids right away. Antimicrobial agents might shorten the duration of diarrhea, which would lead to less fluid loss, which would possibly improve the patient's chances of surviving the infection. Vibrio cholera is resistant to tetracycline and doxycycline. Not all strains, but there have been strains that are resistant. Now, epidemic cholera, Vibrio cholerae 01 has two biogroups, the classic and the L-Tor. The L-Tor is Voges Proskauer positive. It hemolyzes red blood cells and is inhibited by the antimicrobial agent polymyxin B at a concentration of 50 micrograms. Infections have occurred involving other serogroups as well, such as Vibrio cholera non-01. Usually they're milder since they lack that cholera toxin gene. You can get gastroenteritis, also some extraintestinal infections such as cellulitis, ear infections, and septicemia. But the major problem with cholera is this watery diarrhea. Vibrio cholera serogroup 0139 originated in India in 1992, caused widespread cholera cases throughout India and Bangladesh, and is predicted to be the cause of the 8th pandemic of cholera. Now here's just a little saying and picture about Vibrio cholerae to help you memorize some of these things. So, hi, I'm Vibrio cholera. I'm a gram-negative, oxidase-positive, fermenting bacteria. Many of my strains cause cholera, a severe diarrheal illness. I can be epidemic or pandemic, and I love the developing world. I have a high fatality in malnourished populations where people get very dehydrated. To give you severe diarrhea, my strains need to make the cholera toxin. I have lots of strains. Most of my strains that cause outbreaks are type 01 strains, but there are also 139 and non-type 01 strains. You can classify me into Inaba, Ogawa, and Hikojima serotypes if you want, or you could classify me into classical and L-Tor biotypes. You don't see my L-Tor much anymore though. So this came from a website, the Adopt-A-Microbe blogspot.com, and they have some really cute pictures and sayings about all the different microbial organisms. This is a picture in March of 2009. There was a very serious cholera outbreak in Zimbabwe that caused up to 60,000 infections and over 3,000 deaths in a very short period of time. So cholera outbreaks in developing areas can be devastating. So now we're going to move on to the second part of this lecture.